Conrad Waddington proposed in the 1950s that there was something on top of the DNA, something controlling it and triggering genetic changes. Some scientists began investigating if it was possible that a person's environment could cause these changes. This resulted in epigenetics, which is mostly the study of heritable changes that are not caused by changes in the DNA sequence. This suggested that the sequence of the DNA isn't enough to explain cell behavior. Epigenome literally means above the genome. It is a molecular marking system that controls gene expression without altering the DNA sequence. In a sense, the epigenome is the genome's boss. Each new cell has identical DNA, but the epigenome orders some cells to work different than others. It was believed that the DNA in genes were built, and after nurture, there were no changes, except for some during puberty. Basically, they thought that most of the program was determined. However, they discovered that the boss is always watching, and the DNA constantly changes because of a lot of factors. The epigenome is the mediator between the genes and the environment. Epigenetic changes are biological markers on DNA that modify gene expressions without changing the DNA sequence. Researchers have found that lots of factors such as trauma, stress, food, pollution, toxic chemicals, drugs, even exercise, social interaction, and interpersonal relation can activate epigenetic changes. One of the most important factors is nutrition. According to Randy Turtle, food is truly medicine when you're talking about epigenetic changes. By looking at all the factors, we can see the power we can have over the health, since most traits and diseases involve a multitude of gene combinations. Genes are not strictly our destiny. Epigenetic traits can be passed down from generation to generation. However, in some cases, epigenomic decisions can be reversed. Understanding epigenetics can lead to better medical treatments, such as those being developed for cancer and schizophrenia, which consist in reprogramming cells by reconfiguring the epigenetic markers.